Particular structure means appendages. Particular structures means appendages. Some things are found about some of bacteria, not all of them. At first, about capsule and slime layer. Slime layer and capsule. What is capsule? Capsule definition. Extracellular gelatinous accumulation of viscous materials around the external surface of the cell wall of bacteria. Extracellular gelatinous material around of cell wall of bacteria is capsule. This is external surface layer of bacteria after cell wall. The bacterial capsule is a very large structure of many bacteria. Some of bacteria have capsule and some of capsules are very large structure. Like Klebsiella. In bacteria named Klebsiella, capsule is very large. Because of that, in this bacteria, you see big colonies, large colonies of this bacteria because of large capsules and mucoid colony and large colony of Klebsiella is related to its capsule. Sometimes capsule is a polysaccharide layer. It is a polysaccharide layer that lies outside the cell envelope, and it is thus deemed part of the outer envelope of a bacterial cell, and is thus deemed part of the outer envelope of a bacterial cell. It is a well-organized layer, not easily washed off, and it can be the cause of various diseases. It is a well-organized layer, not easily wash off, and it can be the cause of various diseases. It means the pathogenicity or the virulence factor in some of bacteria is capsule related or responsible for pathogenicity, virulence in some of bacteria like Streptococcus pneumoniae or pneumococcus causes pneumonia in human, and the most virulence factor is capsule. When capsule diffuses into the surrounding medium, and remains as loose undermarcated secretion as in Loconostoc, it is known as a slime layer. Yes, slime layer is capsule but is loose, not strong
slime
like capsule. You can see capsule by staining, a specific staining with general microscope. With general microscope, you can see the capsule, but you cannot see a slime layer. You can slime layer in bacteria with electron microscope, EM, electron microscope. Because a slime layer is a loose layer, Function of capsule. Function of capsule. 1. Capsule enhances bacterial virulence by inhibiting phagocytosis. Yes, capsule can enhance bacterial virulence. Why? Because of antiphagocytic activity. Because of inhibiting phagocytosis. Loss of capsule may render the bacterium avirulent. Yes, especially in pneumococcus. If a pneumococcus lost the capsule, the bacteria will be avirulent, not non-pathogen. Avirulent means non-pathogen. Bacteria tend to lose capsules repeated subcultures. When you culture and culture and culture a bacteria, when you repeated culture of bacteria, it may be lose capsules. Yes. Capsule can destroy because of repeated subcultures. Subcultures means culture and culture and culture. Culturing of bacteria in more times, many times. 2. Acts as protective covering against antibacterial substances such as bacteriophages, phagocytes, enzymes, etc. Capsule acts as protective covering against antibacterial substances such as bacteriophages, phagocytes, enzymes. Yes, capsule is a protective agent, is a protective item for bacteria. It can protect the bacteria from antimicrobial substances, bacteriophages, phagocytes, some enzymes. 3. Capsule antigen is specific for bacteria. Capsule antigen means antigen K, antigen K, antigen K or antigen K is related capsule antigen and specific for bacteria. And sometimes it is used for serotyping of bacteria. Serotyping in taxonomy of bacteria. In taxonomy of bacteria, and classification of bacteria. Sometimes we use serotyping. Because of the difference in antigenic characteristics in capsule of some of bacteria, we can use difference in capsule antigen as serotyping method. Capsule antigen is specific for bacteria, and you can prepare a suspension on a slide, and use antiserum related to the capsule on it, and then stain by methylene blue and observe in microscope. In microscope, you can observe swirl bacteria. It means covalent reaction test is positive for pneumonia. Other streptococci is covalent test negative, but Streptococcus pneumoniae is covalent test positive. Some example. In gram-negative bacteria, Escherichia coli or E. coli, Escherichia coli, E. coli, Pseudomonas aeruginosa, Salmonella, and Klebsiella pneumoniae have capsule. Escherichia coli, Pseudomonas aeruginosa, Salmonella, Klebsiella pneumoniae have capsule. And in gram-positive bacteria, Bacillus anthracis, the agent of anthrax. Bacillus anthracis, the agent of anthrax, has capsule. Its capsule, its composition is polypeptide, a polymer of D-glutamic acid. Polypeptide capsule, a polymer of D-glutamic acid. Another gram-positive bacteria like Streptococcus pyogenes have capsule too. Hyaluronic acid capsule. It has hyaluronic acid capsule. Other streptococci. Staphylococcus aureus, Staphylococcus epidermidis. It rhymes Streptococcus epidermidis, not Staphylococcus epidermidis. Staphylococcus aureus have polysaccharide capsule. This slide is capsule staining in streptococci. Streptococci are cocci in chain. Yes, cocci in chain, line chain or short chain, line chain, big chain or short chain. And around the cells, you can see capsule around the cells in streptococci. Yes. About cell wall. About cell wall. Cell wall is rough and rigid structure. Rough and rigid structure, not loose structure. Rigid structure, stable structure. 20 to 25 nanometer in thickness and weighs about 20 to 25% of dry weight of the cell. Two items about cell wall structure, thickness and weight. Thickness and weight. Rigid part is peptidoglycan, which is a mucopeptide composed of N-acetylmuramic acid and N-acetylglucosamine molecules alternating in chains, cross-linked by peptide subunits. In both of bacteria, gram-positive bacteria and gram-negative bacteria, we have peptidoglycan. There are peptidoglycan. Peptidoglycan is a complex molecule. It is composed of two amino sugar, N-acetylmuramic acid and N-acetylglucosamine, and some amino acids and peptide bridge. I speak about it in next slides. The bacterial cell wall definition and functions. The bacterial cell wall definition and functions. The bacterial cell wall provides structural integrity to the cell. The bacterial cell wall provides structural integrity to the cell. The bacterial cell wall provides structural integrity to the cell. Protects the cell from internal pressure caused by the much higher concentrations of proteins and other molecules inside the cell compared to outside the cell. Bacterial cell wall protects cells, is a protective item, is a protective agent for osmotic pressure, high osmotic pressure. Protects the cell from high osmotic pressure. Sometimes osmotic pressure caused by higher concentration of some proteins, and cell wall can control it. 3. The bacterial cell wall differs from all other organisms by the presence of peptidoglycan. Bacterial cell wall differs from all other organisms by presence of peptidoglycan. You can see peptidoglycan just in bacteria, molds, or fungi, yeast, protozoa, human cell, plant cell, animal cell don't have peptidoglycan. Peptidoglycan just found in bacteria, specific for bacteria. Poly-N-acetylglucosamine and N-acetyluramic acid. 4. Peptidoglycan is responsible for the rigidity of the bacterial cell wall and for determination of cell shape. Yes? Cell shape, cuckoo shape, rod shape, spiral shape of bacteria related to cell wall. Cell shape related to cell wall. And rigidity, stability of a cell related to peptidoglycan. And last item, it is relatively porous and doesn't prevent for some substrates to pass through. It is a barrier. It is a barrier. It is a protective agent to prevent for small substrates to pass through. We say about gram-negative bacteria, gram-positive bacteria. Negative or positive means, what means negative and positive? A person, a scientist named Christian Gram, studied about the composition of cell wall in bacteria. Yes, a scientist named Christian Gram studied about the cell wall structures in bacteria. And because of the difference in bacterial structure in bacterial cell wall, he designed a type of staining named Gram staining. Based on this staining, bacteria are classified in two groups, gram positive and gram negative. Bacteria may be conventionally divided into two further groups depending upon their ability to retain a crystal violet iodine dye complex when cells are treated with acetone or alcohol. This is about the method of staining. I explain about this method. This reaction is referred to as the Gram reaction, named after Christian Gram, who developed the staining protocol in 1884. In this image, in this slide, you can see gram positive in left, gram negative in right, gram positive in left, gram negative in right. Both of them have peptidoglycan, yes, in green color. Green color is peptidoglycan. It is thick in gram positive, yes. It is thick in gram positive. You can use, you ask me, positive means bacteria become pigmented? No, not at all. No, no. I speak about positive or negative meaning. No, it's not related to pigmentation. Not at all. No, no. Be patient. I explain about it. In gram positive bacteria, in gram positive bacteria, peptidoglycan is thick layer. In green color, you can see in this slide. And in gram negative bacteria, peptidoglycan is thin layer, is very thin. But in gram negative bacteria, you see another layers, outer membrane, brown color, and periplasmic space, between cytoplasmic membrane and outer membrane. And peptidoglycan exists in periplasmic space. Peptidoglycan present in periplasmic space in gram-negative bacteria. Bacterial cell wall. You can see the thickness of peptidoglycan in gram-positive bacteria. Peptidoglycan, dark color, black color, and plasma membrane in gram-negative bacteria. Outer membrane is thick, but peptidoglycan is thin layer and plasma membrane. Both of them have plasma membrane or cell membrane or cytoplasmic membrane. Cell membrane, plasma membrane, or cytoplasmic membrane are same name. Yes, gram-positive peptidoglycan in this slide is purple. Peptidoglycan purple, and membrane. In gram-negative bacteria, peptidoglycan, outer membrane, and cytoplasmic membrane. Function of cell wall of bacteria. Function of cell wall of bacteria. Shape of bacteria. Morphology of bacteria is related to cell wall. If a bacteria lost cell wall, after that the bacteria have not distinct shape. Then, shape of bacteria related to cell wall. 2. Protection to the cell against osmotic damage. Protection to cell against osmotic pressure, high osmotic pressure. 3. Takes part in cell division. Cell division related to cell wall and possess target site for antibiotic. For example, receptor for penicillins. Receptors for penicillins in bacteria exist in cell wall. Lysozymes, an enzyme lysozymes and bacteriophages carries bacterial antigens. Cell wall possesses target site for antibiotics, lysozymes, bacteriophages, and carries bacterial antigens. Some bacterial antigen exist on cell wall. Difference between gram-negative and gram-positive bacterial cell. Gram-positive, P-O-S-E, not G. Difference between gram-negative and gram-positive bacterial cell wall. This slide is comparison of gram-negative cell wall and gram-positive cell wall. In right, you can see gram-positive. Gram-positive. Inner membrane. Cytoplasmic membrane. In both of bacteria is the same. Cytoplasmic membrane or cell membrane or plasma membrane. It is composed phospholipid and protein. Blue protein. Blue protein means general protein. Phospholipids and protein. In gram-positive, you can see peptidoglycan. Peptidoglycan. Many layers of peptidoglycan in blue color. Many layers of peptidoglycan in blue color. But in gram-negative bacteria, peptidoglycan is thin, one layer or two layer. Yes, one layer, sometimes two layer of peptidoglycan. But in gram-negative bacteria, you can see outer membrane. Outer membrane. outer membrane, like cytoplasmic membrane, is composed of phospholipid and protein and other things. Phospholipids, proteins, and other things. Lipopolysaccharide and other things. Peptidoglycan in gram-negative bacteria is in periplasmic space. Periplasmic space. Gram staining. Gram staining protocol. Left, gram negative. Left, gram positive. Right, gram negative. For gram staining, the first stain is crystal violet. Yes. At first you prepare a slide and fixation with heat and start the staining. The first stain is crystal violet. It is blue or violet for 1 minute. Crystal violet for 1 minute. Both of bacteria became blue. Yes, became blue. After that, you use iodine solution. Iodine or Logol or iodine solution. Iodine fix the crystal violet stain on cell wall. Iodine fixes the crystal violet stain on cell wall. In this stage, both of bacteria are blue. After that, next stage is decolorization. Decolorization. Remove color. Removing stain. Removing crystal violet color. For decolorization, we can use alcohol or acetone alcohol. Yes, for decolorization, you can use alcohol, ethanol, or acetone alcohol, or acetone. Gram-negative bacteria have many lipids, a lot of lipids. Alcohol and acetone or alcohol acetone destroy the cell wall of gram-negative bacteria because of lipids, and these bacteria remove or delete their color and became colorless. But gram-positive bacteria don't have any lipids and remain blue. And last stage, you must stain colorless bacteria. You use safranin. Safranin is red. Or Hussain. Hussain is red too. For 30 seconds. For 30 seconds. For 30 seconds. For 30 seconds. Gram-negative bacteria can absorb the next stain, safranin, and became pink or red. For after gram staining, the gram-positive bacteria are blue or violet color, and gram-negative bacteria are red or pink. Thus, the gram-positive or gram-negative related to outer membrane composition, lipid composition, not peptidoglycan. Not peptidoglycan. You asked me, it depends to peptidoglycan layer? No, it's related to outer membrane layer in gram-negative. It's related to lipids material. For example, lipopolysaccharide, lipopolysaccharide, lipoprotein, phospholipid in outer membrane of gram-negative bacteria. The procedure of gram staining. Procedure of gram staining. Crystal violet, all purple, yes. In this stage, all bacteria became purple or blue. After that, iodine, all purple or blue. After that, alcohol or decolorization, gram-positive became purple and gram-negative became colorless. And next stage, safranin or Hussain. It is red. Gram-positive are purple and gram-negative will be red or pink. Application of gram stain. Application of gram stain. Routine use for primary microscopic examination of clinically significant specimens. It is a routine test in laboratory. When you work with any of bacteria, most of times you do gram staining because you want to know about the type of bacteria. Is it gram positive? Is it gram negative? And you use gram staining. It is a routine test in laboratory, in clinical laboratory, in microbiological laboratory. 2. To characterize bacteria growth from culture. Yes, to characterize, to identification bacterial growth from culture. Yes, a structure of gram-positive cell wall in detail. A structure of gram-positive cell wall in detail. You can see cytoplasmic membrane, cytoplasmic membrane, phospholipid and protein, phospholipid and protein, phospholipid and protein, and peptidoglycan, a thick layer of peptidoglycan, and a specific material like lipoteichoic acid. Yes, you can see lipoteichoic acid. Lipoteichoic acid is a type of teichoic acid connected to phospholipid, connected to cytoplasmic membrane. Yes, you can see lipoteichoic acid connected or bound to cytoplasmic membrane. And teichoic acid or teichoic acid bound to peptidoglycan, not cytoplasmic membrane. And some proteins, yes, in brown, some proteins. Gram-positive cell wall explanation. Cell wall in gram-positive bacteria, usually thick, homogeneous, composed mainly of peptidoglycan, usually thick, homogeneous, composed mainly of peptidoglycan. It accounts for 50 to 90% of the dry weight of the cell wall. It accounts for 50 to 90% of the dry weight of the cell wall. And cell wall contain large amount of teichoic acid. You see teichoic acids, polymers of glycerol or ribitol joined by phosphate group. What is teichoic acids? Teichoic acids are polymers of glycerol or ribitol joined by phosphate group. Peptidoglycan. Peptidoglycan. Peptidoglycan, also known as murein. Thus, murein is another name for peptidoglycan. Yes, murein is another name for peptidoglycan. Murein is a polymer consisting of sugars and amino acids that forms a mesh-like layer outside the cell membrane of most bacteria, forming the cell wall. Peptidoglycan or murein is a complex molecule, complex polymer consisting sugars, two sugar, N-acetylglucosamine and N-acetylneuramic acid. The sugar component consists of alternating residues of beta 4-linked N-acetyl glucosamine and N-acetyl muramic acid. Yes, N-acetyl glucosamine and N-acetyl muramic acid in alternating form. These subunits, which are related to glucose in their structure, are covalently joined to one another to form glycan chains. These subunits, which are related to glucose in their structure, are covalently joined to one another to form glycan chains. Attached to the N-acetyl muramic acid is a peptide chain of 3 to 5 amino acids. Yes, peptidoglycan is a complex molecule. We say about 2 amino sugar and peptide chain 3 to 5 amino acid connected or bound to N-acetylmuramic acid based on bacteria. Attached to N-acetylmuramic acid is a peptide chain of 3 to 5 amino acids. The peptide chain can be cross-linked to the peptide chain of another strand, forming the peptidoglycan. It means in one strand of peptidoglycan, we have peptide chain. In another strand, we have peptide chain. Peptide chain in two strands can connect it with peptide bridge, with peptide bonds. Yes, peptidoglycan. I told you about peptidoglycan. This layer is rigid layer that provides strength to cell wall. Polysaccharide composed of N-acetylglucosamine, N-acetylmuramic acid, amino acids related to N-acetylmuramic acid, lysine or D-aminopimlic acid, and cross-link differently in gram-negative bacteria and gram-positive bacteria. Cross-link is not same, is different from a bacteria to another bacteria. Sometimes this cross-link is direct. Sometimes it's pentaglycine in Staphylococcus aureus. This slide is the structure of peptidoglycan. Yes. Glycan backbone, N-acetylglucosamine, N-acetylmuramic acid in alternative form. N-acetylmuramic acid connected to amino acids, L-alanine, D-glutamic acid, D-aminopimlic acid, and D-alanine. and peptide bridge, peptide poles, peptide bridge in one strand to another strand, in one strand to another strand. This is about E. coli. This is about Staphylococcus aureus. It's a more explanation about peptidoglycan. N-acetylglucosamine, G, N-acetylmuramic acid, M, peptide bonds, and amino acid, L-alanine, D-glutamic acid, D-alanine, D-aminopuric acid, and others. The strand of peptidoglycan and peptide bridge, yes, NAG, N-acetylglucosamine, NAM, N-acetylmuramic acid, and peptide chain or oligopeptide. Thick peptidoglycan layer, pentaglycine cross-linkage in Staphylococcus aureus. Teichoic acid or teichoic acid. What is teichoic acid? Some of gram-positive bacteria have teichoic acid. Teichoic acid. What is teichoic acid? Teichoic acid is polymer of ribitol and glycerol joined by phosphate groups. Some have peptidoglycan, teichoic acid, all have lipoteichoic acid. It means some of gram-positive bacteria have teichoic acid, not all of them. Some of gram-positive bacteria have teichoic acid, but all of gram-positive bacteria have lipoteichoic acid. Yes, all of gram-positive bacteria have lipoteichoic acid, but some of them have teichoic acid. Yes, teichoic acid, lipoteichoic acid, yes, lipoteichoic acid. All of gram-positive bacteria have lipoteichoic acid, but some of bacteria have teichoic acid. And protein. Teichoic acid. Teichoic acid are connected to either peptidoglycan or to plasma membrane lipids. I explain a bit about it. Absent in gram-negative bacteria. Yes, just gram-positive bacteria have teichoic acid. Just gram-positive bacteria have teichoic acid. Gram-negative bacteria don't have any teichoic acids. Function of teichoic acid. The role of teichoic acid in bacteria or function of teichoic acid. 1. Antigenic determinant. Antigenic determinant. Receptor molecule for bacteriophages. Participate in the supply of magnesium to the cell by binding magnesium cation, Mg2+. Sometimes teichoic acids related to antigenic determinant. They are receptor molecule for bacteriophages. And sometimes they participate in the supply of magnesium. Sometimes teichoic acids can regulate normal cell division. Normal cell division. For most part, protein is not found as a constituent of gram-positive cell wall except M protein on group streptococci. Yes. It means some of gram-positive bacteria have protein and some of them they don't have. For example, some streptococci have M protein, R protein, T protein. Yes, M protein, R protein, T protein. Or Staphylococcus aureus. Staphylococcus aureus has A protein. But some of gram-positive bacteria have not or don't have any of proteins in their cell wall. Yes, 10 minutes rest. Peptidoglycan in this layer. 10 peptidoglycan in periplasmic space. After that, outer membrane. Outer membrane. Outer membrane is a complex layer. Outer membrane is a complex layer. It includes lipopolysaccharide. Lipopolysaccharide. You can see lipopolysaccharides molecules in this image. Lipid A, yellow. Lipid A, yellow. Core polysaccharide, and O polysaccharide or O antigen. O polysaccharide or O antigen. Core polysaccharide and lipid A are the structure of LPS or endotoxin. In outer membrane, you see LPS. You can see phospholipid. You can see proteins. Yes, protein, general protein, lipoprotein, general protein. Protein in this slide is distinctive. Protein, yes, lipopolysaccharide, protein, phospholipid, lipoprotein in yellow color. Lipoproteins are yellow, yes, yellow circle, lipoprotein. And porine, a type of proteins. triple proteins. They are triple proteins. Purple, in purple color. Purine, purine, a type of protein in outer membrane. General protein, brown, and lipoprotein, yes. In gram-negative bacteria in outer membrane, we can see three types of protein. General protein, yes, in this shape is brown. Purine, in this shape is purple. And lipoprotein, in this shape is yellow. Yes, three types of protein. Three types of protein. Yes. This is another image about gram-negative cell wall. I put different image to learn more about cell wall structure of bacteria. In this image, outer membrane, phospholipid, purine, yes, purine is distinctive in this shape. Lipopolysaccharide, periplasmic space, thin peptidoglycan, degradative enzyme, yes, in this shape, you can see degradative enzyme in periplasmic space and inner membrane, cytoplasmic membrane, inner membrane, phospholipid and protein, and after that cytoplasm. From above to in. Periplasm, the region between the cytoplasmic membrane and the outer membrane, is filled with a gel-like fluid called periplasm. Yes. Periplasm composition is gel-like fluid, gel-like fluid named periplasm. In gram-negative bacteria, all secreted proteins are contained within the periplasm unless they are specifically translocated across the outer membrane. Yes, in gram-negative bacteria, all secreted proteins are contained within the periplasm unless they are specifically translocated across the outer membrane. And in periplasm is filled with the proteins that are involved in various cellular activities. Yes, cellular activities in gram-negative bacteria related to periplasm, including nutrient degradation and transport. In previous slide, you see degradation enzyme, yes, periplasm related to nutrient degradation and transport because of their specific enzymes, specific material. Lipopolysaccharide molecule is extremely important from a medical standpoint. Yes. It consists of 3 parts. You see lipopolysaccharide. 2 of them are medically significant. 1, 2, 3. 3 parts of polysaccharide molecule, lipopolysaccharide molecule, or endotoxin molecule. 1. Lipid A is toxic part of this molecule. Lipid A embedded in membrane. After that, core polysaccharide located on the surface of membrane. And 3. O antigen, which are short polysaccharides extended out from core. O antigens, which are short polysaccharides extended out from core. Lipid A, the first part of lipopolysaccharide. It is toxic. The chemical makeup lipid A molecule plays a significant role in our body's ability to recognize the presence of invading bacteria. Lipid A responsible for invading bacteria. Lipid A is a chemical agent, a chemical part of lipopolysaccharide molecule. It plays important role in our body because of its ability to recognize the presence of invading bacteria. It is very important for infectious disease causing or gram-negative causing infectious disease. It is toxic in nature. As a result, the LPS can act an endotoxin, causing symptoms like fever, diarrhea, and shock. Some infectious disease related to gram-negative bacteria are the symptom like fever. Yes, most of gram-negative bacteria causing infectious disease have fever, causes fever, and sometimes causes diarrhea and shock. For example, E. coli is a gram-negative bacteria, causes diarrhea. Or Salmonella is a gram-negative bacteria, causes fever, typhoid fever, or typhoid fever, or paratyphoid fever, Salmonella typhi. O antigen. It is composed of carbohydrates, including glucose, galactose, mannose, and some other sugars in varying combinations. The O antigens can react with their specific antibodies. The O antigens can react with their specific antibodies. O antigen is antigenic part of LPS. And sometimes O antigen is used for serotyping. For example, in Salmonella, because of the difference of O antigen in Salmonella, we have a lot of serotype, over 2,000 serotype, 2,000 serotype. in Salmonella because of O antigen, because of O antigen diversity. And the last slide. In this slide, you can see the difference in bacterial cell wall in gram-negative bacteria, in gram-negative bacteria, and compare them. Property. Thickness of wall. Yes, thickness. This is summary of this section. Property. Thickness of wall. Thickness of wall in gram-negative bacteria is 10 nanometer. It is thin. But in gram-positive bacteria, it is thick. 20 to 8 nanometer. 20 to 8 to 18. 20 to 80 nanometer. But in gram-negative, it is thin. 10 nanometer. One layer. Another item. Number of layers. In gram-positive, one layer. In gram-negative, two layer. In gram-negative, two layer. Outer membrane, inner membrane. It's periplasmic layer. But in gram-positive, we don't have outer membrane and periplasmic layer. Just we have cytoplasmic layer. Peptidoglycan or morein content. Peptidoglycan or morein content in gram-positive over 50% and in gram-negative 10 to 20%. Yes, in gram-negative peptidoglycan is low, 10 to 20%, or 10 nanometer, thin, it is thin. Next item, teichoic acids in wall. In gram-positive present, in gram-negative absent. Gram-negative bacteria don't have teichoic acid, any of teichoic acid. Teichoic acid just found in gram-positive bacteria. Lipid and lipoprotein content. Lipid and lipoprotein content. In gram-positive, it's low, 0 to 3%, but in gram-negative is 58%. 58%. It's high in gram-negative. Protein content in gram-positive is 0, absent. In gram-negative, 9%. Lipopolysaccharide content in gram-positive 0, absent, and in gram-negative is 13%. 13%. Sensitivity to penicillin G. Sensitivity to penicillin G. In gram-positive, yes. Gram-positive bacteria is sensitive to penicillin G, but gram-negative, no. And sensitivity to lysosine, an enzyme. Lysosine is an enzyme, a type of enzyme. Sensitivity to lysosine in gram-positive is okay, yes, and in gram-negative is no. Gram-negative bacteria is not sensitive to lysosine. Thanks a lot, don't tire. If you have any question, you can send me, you can message for me, decision or via WhatsApp group. Good evening. Thanks a lot and good luck.